Now the next part seems a little scary, but it's not, okay? These are toll-like receptors, or TLRs for short. This is basically your early warning system. So you have some macrophages, they have an early warning system. They can basically tell the body something's not right, we've got to do something right now. Now toll-like receptors are basically proteins in the membrane of phagocytes. And these proteins, or these receptors, they look for something to bind. What are they looking for? Okay, they're looking for this, PAMPs. Now what does PAMP stand for, okay? Pathogen associated molecular patterns. So what, right? What does that really mean? Think about things that are associated with pathogens and not your cell. Make sense? You have peptidoglycan? You don't have that, do you? No, you don't have that. So we can buy that, right? Do you all have double-stranded RNA, like some viruses? No, we can look for that, right? Do you all have flagella? Mostly not, maybe sperm, but we're not talking about sperm, right? We're talking about in the body. No. Let's see, do you all have LPS? No, that's in gram negatives, right? So anything like that, right? Anything that is associated with a pathogen, you're basically looking to bind that. So those are your PAMPs, make sense? Right. Now what happens when these TLRs, right, bind PAMPs? Well, we initiate some responses. In other words, we can initiate apoptosis. That's basically programmed cell death or cell suicide. We can tell the cell, okay, kill yourself. You're infected with the virus. You need to die right now. Kill yourself. So apoptosis. We can initiate the inflammatory response. I mentioned earlier, we'll talk more about it in detail. If you do the inflammatory response, basically you're bringing in lots of blood, right? Bringing in macrophages to take care of the job. That's what you're doing. You can also produce stimulants of the adaptive immune system. So basically you kind of tell the T cells, okay, we got a problem here, right? We got to do something about it. Now, these non-proteins, I won't hold you to, but these non-proteins do the same thing as TLRs. Here's the difference. TLRs are on the membrane itself. They are on the membrane, right? They're looking for something to bind, something that belongs to a pathogen. Okay? These non-proteins are on the inside. They're on the inside of the macrophage or inside of the phagocyte, but they do the same thing. They will also bind a PAMP. That's probably easy to understand because phagocytes eat things, right? They eat bacteria, and they may have a non-protein in their cytoplasm that looks for a flagellum, looks for LPS, binds to it, and it will still stimulate the same responses. So in the non-proteins, if they find a PAMP, they stimulate the same response. The same being what? Apoptosis, inflammation, or your immune system. Got it? But these guys on the inside, these guys are basically on the cell surface. That's how they work. Okay. Now, I didn't mention interferons. For example, the TLR system, they can also stimulate interferons. Now, what in the world are interferons? They interfere with something, right? Sure. Okay. All right. They basically interfere with viral replication. That's what they do. They're interferons. So far, we have not talked specifically about viruses, right? We talked about worms and eosinophils. We talked about bacteria, but we have not really mentioned how do you really deal with a virus, correct? Okay. You can make interferons. They interfere with viral replication. Okay. Now, interferons are basically proteins, and they're released by the host cell that's actually affected with the virus. In other words, a host cell has a virus. Now, of course, that cell will eventually die. Okay, there's no hope. There's no hope for the cell that's actually making the interferon. But that infected cell is trying to save its neighbors. Make sense? So that cell's going to die, but it's trying to make sure its neighbors don't die. So it's going to make interferons to warn everybody else there's a virus around, get ready for it. Okay. Now, interferons are good in that they interfere with viral infection, but they cause many symptoms associated when you have a viral disease, like fever, like being tired, like muscle aches, like chills, like headaches, okay? So interferons, they're good, but they also trigger lots of bad side effects. So when you get a viral infection, this is why you feel kind of bad. Maybe the interferon doing that to you. Now, there are two types of interferons. You got type 1 and type 2. Type 1 is alpha and beta. Okay? Alpha and beta basically work the same way, but just made by different cells. So their process is the same way, made by different cells. Type 2 gamma, okay, totally different. It's really made by your T cells. Now type 1 interferons, they come early on in the viral infection. They happen really early on. Because the type 2 interferon is made by T cells, remember T cells are a part of the immune system. They are part of the adaptive immunity. They're your third line, right? That's like playing seed. They come in later on in the infection. So type 2 interferons, they come later on, like days later, compared to type 1. Okay, look. So let's go over this and figure out what in the world are these interferons doing. Of course, you know I love pictures, right? Love pictures. Here we go. This spore cell, right, is being infected by a virus. Talked about, well, you all with Dr. K talked about viral infections, right? So this virus is going to fuse with your cell membrane, right, to release its double-stranded RNA, okay? So the RNA is in there, and as long as the RNA is in there, we'll do a translation, right? And we'll make more viral particles. Of course, this cell is going to die eventually, correct? Now, in effect to help its neighbors, this cell senses the viruses inside, and it's going to make interferon. So far, so good? All right. So this cell is after infected with the virus, it's going to make interferon. The interferon leaves that poor cell, there's no hope for that guy, but he's going to make interferon. Now, here's his neighbor. He's good right now, right? The virus has not gotten to him yet, but the interferon gets to him, and the interferon binds to his little receptors there. Now what? When the interferon gets to the neighbors, they know, okay, there's a virus out there somewhere. I need to do what? Protect myself. Make sense? Okay. So what this cell will do is going to make antiviral proteins, so AVP for short. Make sense? Right? Because he knows somebody's infected, but I'm not going to go out like that. Make sense? So this cell that has interferon bound to it will make what? Antiviral proteins. Now right now, these AVPs are not active right now. Okay, so they are not active. Now let's see, later on, let's go back to this guy. He dies, right? Later on, he dies. Why? Because the viral particles need to get out, right? Okay, so he's dead. Now these virus guys are looking for somebody else to infect, right? So they go to this neighbor. They too bind to the membrane, fuse with the membrane, and release their double-stranded RNA, correct? Here's the deal. Remember this guy, he made his AVPs, right? He's ready, right? He's ready. Those AVPs become activated when they bind to viral nucleic acid. It can be either viral DNA or viral RNA, purpose viral nucleic acid. So now these antiviral proteins, they are now what? Activated. They bind to it. Now what do? When they're activated, they do two things. Okay? Think about it. What do viruses need to do? They need to do what?
Translation.
Once the RNA is in the cell, we gotta do what with the RNA?
Translation.
Okay, so we gotta make mRNA, right? And we gotta translate it into protein, right? So shouldn't we stop that process? Shouldn't we stop viral translation? Shouldn't we do that? So that is exactly what you are doing when you make antiviral proteins. You stop translation as well as transcription. You stop both processes. You stop transcription, which is basically making mRNA, and you stop translation, basically making mRNA into protein. You stop both processes. Now, of course, the cell is affected too, right? It's not selective. Keep that in mind. It's not selective. If you're not making viral mRNA, you're also not making your own mRNA, right? It's not selective. If you're not making proteins for viruses, you're also not making your own protein. Here's the difference. Even though the cell is affected too, it only lasts, this process only lasts for about 3 to 4 days. It's long enough to get rid of the viral infection without this poor cell dying in the process. Got it? So that's how interferons work. So that poor guy's dead, right? But he saved his neighbors, right? The neighbors were prepared. They were ready for it. Now, interferons, they are non-specific. They work against viruses, but any virus, okay? Any virus can trigger interferons. This table summarizes type 1, right, alpha and beta, they work the same way. So what I just talked about, it can be alpha or beta, basic difference, where they come from, okay? Type 2, again, comes from your T cells. And what they do, they actually activate macrophages. They activate macrophages so they can go out, find invaders, and eat them up. Of course, this happens later on in your infection. Why? Because the T cells are doing it. T cells are what? Third line of defense. They happen later on. If the second line fails, then you go to third line. Okie dokie, complement. Now, what do you have to understand about this complement system? So it's complementing your immune system. It's basically a series of proteins that do something, okay? That's simple as that. So complement, they are a series of proteins that do something. The end result of making complement proteins, okay, is basically killing your invader. You basically punch holes in your invader. You cause the cell to lyse or break open. Now the cool thing is this, to help you memorize what complement does, complement forms a membrane attack complex, or MAC. So think of MAC attack, real quick, right? MAC attack. If you can remember that, that's exactly what complement is doing. Complement proteins make a MAC, and MAC attacks cell membranes or cell walls of bacteria. They literally punch holes in bacteria. They punch holes in them, they just die. So think of MAC attack. That is the end result of complement proteins. They punch holes in the invaders. And there's 3 ways to activate this complement process. The classical pathway is activated when there's antibodies present on your invader. So that's the classical pathway. When you have an antibody literally attached to your invader, you trigger the classical pathway. But keep in mind, they all have the same result, okay? They all end up with making a MAC attack. The alternative pathway, it binds to parts of the invader, so anything like LPS, for example, or an endotoxin, these are things on your pathogen, they bind to that, but the result is still the same, MAC attack, same result. The third pathway is the lectin pathway. It is looking for sugars that are on your invader that's not present on your cells. So when they find a sugar, particularly mannose, because our cells don't have mannose, we have glucose, lots of glucose, or our cell membranes. We do not have mannose. But some bacteria, some fungi, they actually have mannose on the outside of their cell wall. Well, if those complement guys find mannose, it triggers the whole process.
What's the sugar called?
Mannose.
M-A-N-N-O-S-E.
Mannose, it attaches to the sugar, and it starts the whole process. We're getting the same results with a MAC attack. You just got 3 different ways of doing what the same thing. Killing the invader by literally punching holes in their membrane. That's how you take them out. This one is really complicated, but it shows you what you're doing, right? Antibody for classical pathway, alternative, some endotoxin, lectin pathway, mannose, but the result is still the same. MAC attack, cell lysis, cells dead. Okay. Now this is like, oh my gosh, okay. Real quick, of course, do you have to learn all the, there's like 10 steps there. I'm not going to say learn all 10 steps of the complement pathway. Probably not, right? No. But understand this, it's like a cascade effect. It's like an avalanche. This is really the classical pathway. This guy's a pathogen. Looks like, see that guy? That's an antibody, right? Binding onto antigen. Okay, great. This guy is a part of the whole process. So complement is basically lots of proteins. C1 just starts everything off, okay? If C1 finds an antibody, he binds to it, it just starts the whole process, right? The whole process ends in what? Okay, at the end of the day, right? You make a what? MAC attack, right? At the end of the day, okay, you do this. All of these guys are complement proteins, and they form on that cell membrane, and they punch holes, okay? And if you punch holes, this guy's going to die eventually, makes sense? That's the MAC attack. Here's an actual picture of MAC attack. All these guys there are what? They're holes, right? You're punching holes through that pathogen, and that guy will die, right? It's called complement, or the complement pathway. Basically, make MAC attack. Make holes in your pathogen, they die. That's it. does your body deal with a Mac attack? Because I tell you what, this cascade process, this common process, is not specific. So this Mac attack could end up on your cell. It very well could. Okay? So how do your cells deal with a Mac attack? All right? Your own cells, they have proteins on their membrane that deactivate the Mac attack, so they don't end up dying in the same way. Because those guys, they can fall on your cells too. And if they do, they're going to start punching holes. You have proteins on your membrane that deactivates the Mac attack, so you don't go out like that. Okie dokie. So, last two things, inflammation and fever. Basically, inflammation is non-specific, responding to some sort of tissue damage, okay? You basically have lots of things going on real quick. Of course, you know signs, right? Redness, right? Swelling, warmth, and pain. All that's what inflammation tends to be what localized to. So keep that in mind. Because fever is all around, right? But inflammation tends to be localized. Okay? You have two types. You can have acute or long-lasting. So think of long-lasting, which is chronic. Think of things like arthritis, you know, lasts a long time, can really damage tissues overall, okay? But we're focusing on acute because acute is typically dealing with some type of disease, like an infection. It's quick, and it takes care of the process really easily. Chronic is really with arthritis, a long-term inflammatory process. Okay. So we deal with acute, right? Acute means what? Develops quickly, short-lived. It's typically beneficial for two reasons. A, it's real quick, short and sweet, but it helps to take care of what's causing the actual inflammation, right? If there's some staph in that area, it helps to deal with that staph in that area. Now, what's really going on in inflammation? First of all, these two things are happening. First of all, dilation of blood vessels, which means they get bigger, right? And if blood vessels get bigger, they bring in what? More blood, more nutrients, and also some macrophages, probably some phagocytes. Now here's the deal. When you have dilation, that's how you get the redness, right? That's where you get the heat. It's from the dilation from the inflammation process. Likewise, you get increased permeability of blood vessels, which means instead of them being tight together like this, they kind of open up a little bit, which means you can allow macrophages to kind of get in there and do their job. But when you open up the space between cells, fluid leaves the blood vessels, right? And fluid gets into the surrounding tissues, correct? And that causes what?
Swelling.
Right, edema, swelling, makes sense? So that's why inflammation, you got what? Redness, warmth, and swelling. It's a result of what? Dilation and increased permeability of the actual blood vessels. Now, all of this, whether it's dilation, first of all, dilation helps with this process, tissue repair. So when dilation brings in fresh blood, fresh O2, it helps for that tissue that was damaged to repair, correct? Increased permeability helps with this process here, right? Bring in phagocytes, right? Bring them in, help them to get out of the blood vessel, into the tissue itself, and eat whoever's there in the first place. You don't need to go over this picture. This is showing you what before and after, right? So before dilation, after dilation, okay? Bigger vessels, more things can get through. That's all that's trying to show you. All right? This one is showing you what increased permeability. See how these guys are real close together? Nobody can get through. Increased permeability. Okay, this guy can squeeze out, right? That's a monocyte. He's going to be a macrophage. He's going to walk out of there and eat whoever is causing the process. But of course, fluid can leave too, right? That causes your swelling. Now, is there anything I want you to know on this slide? Yes, there is, okay? You need to know the chemicals in your body that causes the dilation and causes the increased permeability of those vessels. You need to know that. Because you probably take drugs that combat those chemicals. You have to understand how it works, correct? In terms of increasing the diameter of your blood vessels is histamine, okay? So histamine causes your blood vessels to dilate, which brings in more blood. So that's why you take antihistamines. It blocks histamine. Now, in terms of increasing permeability of the blood vessels, you've got leukotrienes and prostaglandins. So prostaglandins and leukotrienes, they increase the permeability of the blood vessels. So whenever you take aspirin or ibuprofen, they work against prostaglandin.
Increases permeability.
Right, so those P guys, they increase permeability. When you take ibuprofen or aspirin, they work against prostaglandin. That's their job. So whenever you take ibuprofen, that's what you're doing. It's exactly what you're doing. You're trying to turn off prostaglandin. Okay. We're almost done. Ooh, early too. Here's your last line, right? When all else fails, right? Turn up the temperature, right? When all else fails. You done all the above, right? Cook it. Fry it. Turn it up. Fever, okay? So fever is basically your body temperature that's over 37 degrees Celsius. How does it work? Okay. Your body is responding to something. It's responding to a pyrogen. Pyro means like hot, fire, right? So what are pyrogens? Pyrogens can, this right here. Pyrogens can be basically the bacteria itself. You got bacteria, they can be a pyrogen. Anything that stimulates a fever, it's a pyrogen. Okay? Or cytoplasmic contents from the bacteria when they're broken down, or the antibody-antigen complex. Again, antibody with the antigen, that's going to be chapter 16. That's going to be very specific. But these two guys can be very general. These are pyrogens. They basically stimulate increased temperature. Now, how does this work? All right. Now, remember prostaglandin? Remember that guy? He was the same guy who increased permeability of your blood vessels. Okay. Well, prostaglandin also works in this process too. It helps to turn up the temperature. Now, let's see. Where's our number one? Okie dokie. So, you got chemicals secreted by blah, blah, right. You got pyrogens, right? They make their way to the hypothalamus. So the hypothalamus, that's your thermostat. Okay? It regulates your body temperature at 37. If the hypothalamus senses something's going on, like we got some pyrogens that made its way up there, it's going to turn up the thermostat. Now, how do we turn up the thermostat? Well, we got to make prostaglandins. Prostaglandin will also help to turn up your temperature. Now, what does prostaglandin do? This is why, again, you take what? Ibuprofen, right? And aspirin to do what? Lower the fever, right? If you can get rid of prostaglandin, you bring your temperature back down. Make sense? So what is that doing for you? Well, prostaglandin will do all these things, right? It will help to increase nerve impulses, right? So that you kind of increase temperature, okay? You'll be shivering. You have a higher metabolic rate. All these things increase temperature, right? That's the job, right? Increase fever. Also, blah, blah, blah. Vasoconstriction, basically, instead of the blood vessels dilating, they do what? Constrict, which means you maintain the blood within the body, away from the limbs to warm up the body. That's why you feel chills, right? You feel chilly because most of your blood is down here, not in the extremities, makes sense? That's why you feel chilly when you have a fever. Look it up. That's basically all there is to it. Now, of course, some people say what? Let the fever run its course, because what you're really trying to do is what? Fry the pathogen. Right? Trying to kill it by cooking it to death. But also, if the fever is too high, you also start killing your own cells too, right? So usually, if you do have a fever, it's best to take aspirin, right? It's best to take ibuprofen, right? Because you want to get rid of what? Prostaglandin, makes sense?
Oh, that's why we watch the temperature to make sure it doesn't exceed a specific
right. Range. Depending on your age, right? You don't want to go over a certain number. Low grade fever like 99, no big deal, right?
I go to 100.
101 is where, okay, that's too high, right?
Yeah.
Too high, take something for that immediately. Okay. Look at this. Let's see. Now this table does a nice job of summarizing everything we just talked about today. So I would cut that one out of your textbook, put it probably in your bedroom somewhere like the alarm clock, for example, put it there. That will help you for your test, because some of these questions on your test, about 10 of them will come from chapter 15. There are no surprises. Everything I said today, you should know, correct? That's it. Now we are done. Now, do you have any questions for me in terms of this chapter? Chapter 15, innate immunity. Well, I wish you all a good weekend. Remember, vote before Tuesday, because Tuesday is test day, but it's voting day too. So vote before you come in for your test. Y'all have a good day.
Chapter 15 quiz tonight?
What's that?
Quiz tonight?
Yes! Quiz tonight! Yes. Chapter 15 quiz. Yes, tonight. Thanks for reminding me. Yes, absolutely. Absolutely.
I don't know how I broke that chair.
Oh, you did?
Yeah.